Okay, good evening, everyone. And I hope you have watched the videos and about transgenic animals. I will today summarize about it. And the important thing is, first thing, of course, by definition, what is meant by transgenic animal is it can be taken from transgene, which means this gene is something foreign. Okay, and if you put a gene into the genome of an organism, then it is transgenic. With that, right now it is transgene, or you can also say it as any transgenic manipulation, or genetic manipulation done can eventually also become the transgenic animal. The point is there should be a genetic manipulation, and that genetic manipulation should be present in all the cells of the body, and which mean that it should be also heritable, right? That is the meaning of the transgenic animal in a way. So before going further, I would just want to discuss something like this. Say, for example, there is a genome because it's haploid. That means one is from paternal allele, the other one is paternal set, one is maternal set. And assume I'm describing a genetic locus, something like this. Okay, gene A and gene B, and it also has another copy, gene A and gene B. This is one organism. We would want eventually to have an organism, say in this case, I want to put another gene between A and B, right? A, B, these are the actual ones. Within that, we put a gene X. Okay, gene X is put. And all the cells until then had this kind of organization. And once a transgenic animal is made, it should be homozygous. And the second one, it's not necessarily that everything should be homozygous, but that is what we are aiming for, homozygous. And the second thing is, the important part is that all the cells should have it, not just a small some parts, and not the whole organism. This gene should be present in all the cells of the body. So we cannot directly get those things. Okay, so there are two ways of how you do it. One is the direct method, and the other one is the, you can say, embryonic or engineered stem cell method. In the direct method, you take gene X, and you put it in directly, and hopefully it will integrate. And those that have gotten integrated, you will take them, and then you will allow, because we are only giving only one copy you might integrate. We should make it homozygous. So we should then try to perform inbreeding and keep inbreeding until we get a homozygous one. The engineered stem cell method is a little bit indirect. So because we take the embryonic stem cells, and then in that embryonic stem cells we make this construct A, X, and B. Okay, we make these the genetic manipulation, and those that are positive we will take and we will put it into a blastocyst, which will give rise to a chimera. In the chimera, we will take the chimera and try to further analyze them, because we will allow it to mate with parent type and try to get the pups. Out of those pups, we will maybe we will get the first founder transgenic. Chimera is not a transgenic animal yet. Okay, we will discuss about this, but I just want to give you the overview. And once the founder is there, we will allow it to for inbreeding, and then upon inbreeding, we will select for those that are homozygous and transgenic, right? So we'll just quickly, I would want to focus more on the engineered embryonic stem cell method. We take embryonic stem cells, we do the genetic manipulation here. And here we are actually doing the embryo manipulation. And then we get the founder, and then we are inbreeding and then to produce these things. So the important thing that we have to remember is these two parts, genetic manipulation and embryo manipulation. The rest of the things are actually little easy. Okay, I will just see and this is the something like the embryo manipulation, and then we have the characterization, so that we know which ones are homozygous and that we have established the transgenic animal or not. Okay, this is the microinjection method, that is the direct microinjection method, and the other one is engineered stem cell method I was just describing. Okay, I mean these are the same things exactly as that are present in the video. So what I want to go in is to discuss about the other method, that is the engineered embryonic stem cell method. Okay. Excuse me. Yes, this is where I want to discuss. So the first thing I want you to understand what is meant by positive selection and what is meant by negative selection. We already must have discussed, but it is worth repeating, and I will do that. Positive selection means presence of gene will allow growth. That means if the cell did not receive that particular gene, the cell would have died in the selective conditions. In the negative selection, it is presence of the gene, the particular gene that we are actually using it, will cause the death of the cell. That means if that particular gene is present in the cell, in those conditions which we are going to put, the cell would die. Say, for example, I have taken cells and I put in neomycin phosphotransferase, okay, which is a resistant genes for neomycin. You transfected it, and you did this to many cells, but not all the cells will get start growing in them on nutrient plates, I mean growth medium. These are animal cells. So those cells that have received NPT gene, neomycin resistance gene, will grow. And you might have taken about 100 cells, say, for example. The rest of all the 97 cells actually died because they were not, because we have used neomycin in the growth medium. So those cells that have the neomycin phosphotransferase gene would survive and the rest would die. So that is the positive selection. As I said, the presence of gene is allowing it to grow, right? Positive selection. In the negative selection, say, for example, we give thymidine kinase from HSV, herpes simplex virus. If we give to cells, several of them, similar setup, everything, except that now we are growing the cells in something like ganciclovir. Ganciclovir is a prodrug, which means if it is acted upon by thymidine kinase, it will form toxic compound, and the cell that has this toxic compound will die. Now, when we do this transfection here, thymidine kinase gene into several cells, maybe say again, we have taken 100 cells and say some 97 cells have got it, or 95 cells have got the thymidine kinase gene. How many cells are left? It's only 5. Only 5 of these cells, 5, we will only be able to find 5 colonies, because the rest of the cells that have gotten thymidine kinase, in those cells, the gancyclovir, when it is taken up, it would cause the gancyclovir will be converted to toxic compound and the cell dies. These are the cells that are growing because they don't have thymidine kinase. So that is the negative selection. Presence of gene will cause the death. That means those cells that did not get the gene will survive. Therefore, we call it as negative selection. Okay, we are going to use both of these three cases. There is also one more thing I want you to also remember. I hope you must have seen, if you have seen the videos, you should know the basics of homologous recombination and also the site specific recombination. So when we are making genetic manipulations, we have to be very careful. In the sense, as a setup, I said there is gene A and B, and I want A and I want to integrate X here, and B is there. That means I want to incorporate X between A and B and nowhere else. Usually the scientific statement will be very, very clear. So the statement here is integration or gene targeting, you can say integration of X, gene X, between, or you can say it as within the intergenic region of A and B, between gene A and gene B. That's not sufficient. We also have one more clause like thing, which says, and nowhere else. Right? If you just stuck by this, it may be here, but it might also be somewhere else as well. We don't want anything like that. Why did we choose just A and B? First of all, we might have had characterized that if we incorporate X here, the intended effects will be only because of the X being here, and there would not be any becoming cancerous or other abnormalities. That's why we have chosen it. That means I'm trying to highlight that if X or any of the gene that we are trying to put gets integrated randomly into any place, it might actually cause a lot of problems to the cell or eventually after growing into an organism. So we should have chosen a good location where we want the gene to be incorporated. So here, theoretically, we'll take what we want the gene X to be between A and B. That is safe for us. And there should not be anywhere else. For this, we have to use two things. I'm trying to do it differently than what is already in the video. So you can also watch the videos, which is actually elaborate and in detail. So I'm trying to explain because maybe you might not have looked at it. And so here is gene A and gene B. If I want to target gene X between them, then I should have the gene construct I'm drawing in the blue. I can use homologous recombination. Homologous recombination, if you remember, is based on sequence similarity or complementarity. That means there should be common sequences between these two DNA molecules. Only then homologous recombination can occur. Okay. So what we are doing is, this is the incorporated DNA. This is the target locus. and here we are going to put gene X. How did we construct this? The DNA that we want to transfect is by something similar to that of cutting and joining type of things. There are also PCR based methods as well, right? So now this is the genome of the cell, and we should be able to incorporate. So as I said, first thing is to incorporate X in the in between, within the introgenic region between A and B, and nowhere else. How can we ensure that it is nowhere else? And second, the other important thing is, say for example, we have taken 100 cells and we did the transfection, and maybe out of them 4 out of 100 cells actually got the DNA. How do we figure these out? There should be some selectable marker, right? X, if unless X may not be having a property that is easily visible, and so in that case, how do we distinguish these cells and get rid of the remaining 96 cells? That is where we use positive selection. What if I put neomycin phosphotransferase gene here, NPT? There is resistance for neomycin. If I have grown these cells in with neomycin containing media, the rest of the 96 cells would have died. Only these 4 cells would have survived. Positive selection, right? That is one of the things we should be able to select the gene, select the transfectants. There is a problem, however. What if, say for example, I will draw the genetic locus as it was. I'm trying to draw this one here. Okay, A and B, and assume that this whole DNA construct that I'm showing here got integrated somewhere else. A, X, NPT, and B, right? B prime, small fragments. It did not get incorporated where I want to. Somewhere in the genome, it got incorporated by non-homologous mechanisms. It is always a possibility. If I grow these along with them, will they grow or not? Of course, they will grow. Why will they grow? Because neomycin resistance gene is also present in the genome. It's not in the intended location, it is somewhere else, but it is still there, so it will grow in here. So that means we have two things, or how you see is also important. We should be able to select, first thing is we should be able to select transfectants from non-transfectants. Sorry, non-transfectants. That is the first goal. Then among the transfectants, there are two categories. One, those that are integrated through homologous recombination, and those that have incorporated by non-homologous recombination. We should be able to distinguish these two, right? For this selection, for selection of transfectants from non-transfectants, we are going to use positive selection. And to distinguish homologous recombinants from non-homologous recombinants, we should be able to get the we should be able to use negative selection for it. So say for example, the ideal situation would have been that okay, I will just use this slide that is actually indicating that. There are also more things. What did we establish in the first case? I want X to be incorporated between A and B, nowhere else, and nothing else. In the sense, no other genetic manipulation should have been there. But if you notice here, because we want for positive selection, we have used NPT. We have incorporated NPT into the genome. However, we don't want NPT, nothing else, right? So we have to get rid of NPT, and for that we can use we can use site-specific recombination. I will also still use the my diagrams so that you can probably see how you can draw your own pictures. So how to separate? So the DNA construct is going to be like this: A, and then here we are putting gene X, and we have NPT for positive selection, and then we have gene B. So there are some more things that can be. We have two things to do. First is we should be able to separate homologous recombinants from non-homologous recombinants. To eliminate the non-homologous recombinants, we can use thymidine kinase. How we do that is we will put thymidine kinase gene here. Okay, there are two fates. If this DNA construct integrates through homologous recombination, it will be A, X, NPT, and B, right? So as is discussed here, as I discussed here, homologous recombination will only happen within these boundaries, homology. If I put thymidine kinase gene here, it's not going to get incorporated if it has undergone homologous recombination, right? So, So if it is through homologous recombination, if incorporation has happened, then thymidine kinase does not get integrated. Instead, if there was a non-homologous integration, then the whole bit from here till here will get incorporated. NPT, B, and also thymidine kinase. These are non-homologous recombinants, right? So what do we do now? We can take these two different types of cells and grow them in plates containing a compound or gancyclovir. There are other compounds also with other different names. So the homologous one does not have thymidine kinase. It is TK minus. Whereas the non-homologous recombinant, it is TK plus. Therefore, when we have gancyclovir is going to be converted to a toxic compound by thymidine kinase, and therefore these cells will die. So we are able to, using negative selection, we can eliminate all the non-homologous recombinants. Our problem. We want X, incorporation of X between A and B and nothing else, right? But we used neomycin phosphotransferase. We need to get rid of it. So how can we get rid of it is a question. There we can use site-specific recombination. I hope you remember I made you do homologous recombination exercise before we started this course. And similarly, we also did for site-specific recombination. In site-specific recombination, these are referred to as recombinase recognition sites. They may have small stretches of sequences with a middle region that is called as the crossover region. So recombinase, the enzyme usually recognizes these serine recombinases, tyrosine recombinases. They are there. They recombine and then they exchange the DNA. That means they would cut the DNA somewhere like this, and then they will rejoin them. Let's see how it is. What will the consequences shown here? I'm just drawing the arrowheads so that it's easy for us to understand. I put them facing each other. There is also another way you can put it, where they are both facing to the same side, right? How to resolve this, the recombinase, the through site-specific recombination is, if we take this one, we have to put it always recombinase. What recombinase does is it will put the recognition sites in the same orientation like this, and then what it does is it will cut the DNA here. Okay, let me erase all of it. And then what it does is it will exchange the DNA molecules like this. Okay, and if you linearize it, it will be just DNA, but in the middle part there has been change. So we are going to solve these two, and I hope you will be able to follow. So let's see. I will try to have it a little bit different. I will write the gene names. Say A is not a good one. P, and then I'm going to put this way Q, R, and I'm going to put this way and S. So the first goal, as I said, is that we have to put the recombinase recognition sites in the same orientation. Both should be facing the same side. So if I want to do that, I should bend the DNA like this, right? We have P, then we have the recombinase recognition site facing Q, then we have R, and we have the recombinase recognition site facing R, and then S. So now recombinase would have cut this DNA like this and would have rejoined differently like this. Okay, now let's try to pull it straight. We have P. P is continuous with R, and that is continuous with Q, and that is continuous with S. And of course, we will have the recombinase recognition site. This is the original DNA where we had P, Q, R, S. And upon site-specific recombination, now we have P, R, Q, S. What has happened? This region, it got inverted. Okay, that is how you solve this. This is not so useful for us for the system. How we want to remove the neomycin phosphotransferase, it's not going to be useful. So we're going to see what will it be if we had it in a different orientation. So here I'm going to put it in the same orientation. P, Q, R, S. So I'm going to bend it like I did before. This is P, and then we have Q, then we have R, and this is facing R and S. One of the rules that I told you is that both the recombinase recognition sites should be in the same orientation. As I drew here, it is not in the same orientation. So I have to redraw it. So how do we rearrange it? Is to push it further one more round. We have Q here, then we have R, and we have this, and then we have S. Now let's see what happens upon the recombination. The recombinase would cut the DNA like this, and now it is going to join P and S like this, and this one like this. If we keep these two molecules differently, we would get P, a recombinase recognition site, and yes. And the other one would have been something like this. R and S. What has happened? The original sequence was PQRS, but now we have P and S in one DNA molecule, R and Q, yes. R and Q as a different molecule. What has happened? Deletion has happened. So we can use this one to get rid of the neomycin phosphotransferase gene. That is what we have here, right? We don't want it. But we had to put it because we wanted to select transfectants from non-transfectants. So we need to remove it. Therefore, we need to put it this way. Why am I putting both these recombinase recognition sites in the same orientation, that is when you will get rid of the neomycin phosphotransferase gene, right? Upon non-homologous, sorry, upon site-specific recombination. So these are, there is a, you will, if you are going to read a lot of papers where genetic manipulation has been done, you will find this CRE and LOXP. LOXP is the site name, this the recombinase recognition site name, LOXP sites you call it. CRE is the recombinase. This is well standardized and many people actually use it. This is for eukaryotic. In prokaryotes also you have different, there are other recombinase sites as well, but this is one of the popular ones. Principle will remain the same. Okay, I think we are now in a state where we have, I hope you can understand why we have each of these things. If you understood that, then we are good. Why do we have A and B? Because we want the integration of X between A and B. So we are putting the homologous region so that homologous recombination would occur. Then X is the transgene that we want to introduce into the genome. So we have it. Why do we have NPT? It is for positive selection, because we should be able to differentiate which ones are the transfectants, which ones are not the transfectants. Using positive selection, I mean NPT, we can separate them. And there is a problem is that we may have homologous recombination, but we may also have non-homologous recombination. For that, we are going to use thymidine kinase as for negative selection, because thymidine kinase, when acts upon ganciclovir, will convert ganciclovir into a toxic compound. And if there are any non-homologous recombinants, they would have inherited TK, so they would have died. Okay. And then we have the last part is the recombinase recognition sites. We put them both in the same orientation, because only then we will have deletion, otherwise not. Okay, this is the gene construct. So we have everything here. Gene X, you can see, you can do deletions, you can do insertions, many things can happen. Okay. So this is random insertion, what I meant by non-homologous integration or recombination. This is gene targeted insertion, that is by homologous recombination here. Homologous recombination or non-homologous recombination. There is thymidine kinase gene. Thymidine kinase gene will not get incorporated if it is the case of homologous recombination. In the case of non-homologous recombination, however, we will also get the incorporation of the thymidine kinase gene. And so we can first select using positive selection. We should be able to select recombinants from non-recombinants, whichever has received DNA or not. So the recombinants, when we use neomycin, all those that receive the DNA will survive and the rest will perish, like these ones. Now we have to separate the homologous recombinants from non-homologous recombinants, right? So these are the non-homologous recombinants, which have the blue color indicating here the thymidine kinase gene. So when we use ganciclovir, and those cells will die, and only those cells which have the transgene in the correct location between A and B, for example, will survive. So now what do we have? We got engineered embryonic stem cells. Right? We have modified them. We are a long way to get to transgenic animal. We didn't get this. These embryonic stem cells can give rise to all types of tissues, but not an whole organism as such. Okay, we will discuss those. You might have already studied, but even then, if not, we will still discuss it some other time. So now we are, if you recollect, I was trying to make it into several parts, right? Where is it? There are 3 different parts. Okay. First one, as I said, the genetic manipulation. And the second part is the embryo manipulation. Embryo manipulation, to get, we have cells. From cells, we should somehow be able to get the organism. So what we do is we take a blastocyst. Blastocyst, I mean, is a zygote will grow into morula, and the morula will grow into a blastocyst. Blastocyst is something like this, an outer layer of cells, and then you have some cells inside. This is called as inner cell mass, and that is the source of embryonic stem cells. Every time if we have any embryonic stem cells, it must have come from the ICM of some blastocyst. Okay, all the tissues in our body come from this ICM. So ICM is similar to or is the source for embryonic stem cells. So what we are going to do is we have the engineered embryonic stem cells, right? We are going to take the engineered cells, okay, manipulated cells. Oh, here, say for example, we took the embryonic stem cells, we made the genetic manipulation, this part. We have selected and we got the engineered cells, and now those cells, the engineered cells, are going to put into a new blastocyst, the recipient blastocyst. So through microinjection, cells can be put inside, and then they will put it into a mouse, a female mother. It is a surrogate, and there is a procedure for it by which is called as pseudopregnant mouse. Pseudopregnant mouse, in the sense they will allow this female to mate with a vasectomized male, so that physiologically, because of the activity, the female will have several hormones and others secreted, because of which it will be ready to receive and conceive and hold on to the embryos. So physiological preparation or hormonal preparation is what is done. So they put these kinds, several of them, several embryos, manipulated ones, into the surrogate mother, and then they will allow it to give rise to pups. And then we have to perform molecular. This is the first transgenic founder. In the sense, it is still too direct here, right? Say for example, I'll try to use this cell. Sorry, I'll make a slide here. So we have a blastocyst. In some cells, within the ICM, I have two different types of ICM. One is colored, and the other one are white cells. If this gave rise to a mouse, okay, some parts of the body will be made from these blue cells, and some parts of this body are made up of the white cells. So actually, we will have one single organism developed from two different embryos, and that is a chimera. In all of our mythology, we have an elephant head and a human body. Greek mythology have Hydra, many other types, right? There, the intention is that one individual made from two body parts from two different species. When we speak here in biology, we are talking here as chimera is about any organism that is developed from two different embryos. How is it from two different embryos? The white cells have come from a different embryo, and the blue cells have come from the different one. Here we took first donor blastocyst, from which we took the embryonic stem cells or ICM, and then we did all the genetic manipulation, and then we are putting it into a different blastocyst, that is the recipient blastocyst. Okay. So the embryo that comes here is the chimera. All individuals have, an organism has two parts, right? Somatoplasm, that is the whole of the body, and then the germplasm, that is what gives rise to gametes. When we were talking about transgenic animals, we also discussed that it should be inheritable. What if the whole of the organism is made up of the blue cells. Say, assume blue cells are the manipulated ones, but its germplasm is made from white cells. Okay, if that is the case, the trait is not going to be inherited. Okay, so what we should be doing is, because we have to identify whether which of these chimeras is actually useful to us. So they would allow, we are assuming that the blue one is the manipulated one. Okay, blue one is the manipulated or transgenic type. We will allow the female, this mouse, to mate with a white one and then see the pups. There are three possibilities. Pup can be completely white, pup can be completely blue. There are only two possibilities, either completely white or completely blue. Okay, assume this is also a mouse. It's ugly, but okay. Now, if it is completely white, that means the germplasm of this individual is made from white cells, and that's why it is given as it is white. If it is completely blue, then it would mean that the germplasm is made up of the blue kinds. Okay. In one litter, that is at a time when the birth is given, both are possible. Because in which case is it possible? If the germplasm is made both by white and blue cells, okay, both contributed, some gametes are in white, some are in blue, then you will get different types. What is our interest? Is the blue one. That is what we will take. It is heterozygous, and then we will perform inbreeding. That means you will take several of these heterozygous founders, and then you will allow inbreeding so that you will get a homozygous one. You will perform in the inbreeding experiment on them until we get homozygous individuals. Okay, I'll just ask you this question, and if you can answer, it'll serve a lot of purpose. Can a chimera form in this stage? Yes. The experimental setup is a chimera is allowed to mate with a recessive gene or the wild type, and then we are saying one white has formed, one completely blue is formed, and a chimera is also formed. Is that possible?
It should be, sir.
Why is that? How is it? Yes, how is it?
Like, say, for
Please.
example, there are bar bodies and certain genes get activated at certain cells. So there is a very good possibility
Um,
for us to have patches.
you're right in that sense if it is on X chromosome, right?
Yes, sir.
Otherwise, bar body doesn't make any difference, isn't it? Assume we have put it in. We usually when they don't do it on the sex chromosomes. They do it on the other ones because we need to have a male and female, right? So sex chromosomes usually will be avoided. Yes, try again.
Okay.
So if this one has made gametes, okay, the gamete will either be white or blue. This individual may produce both of them, but a gamete will be either white or blue one. Whereas the male, assume this is a male, and it would produce only white ones. So if you see that this will be white, and if this and this one are fused, you would have one white and one blue. And assuming the blue is dominant, this will give rise to a blue pup, whereas this one will give rise to a white one. Okay?
Hmm.
Yes?
Yes, sir.
Okay. So these are several other same things. I mean, on YouTube, you can find, I used to have them, now I don't have it here. There are fantastic videos which will demonstrate how which will demonstrate how the embryo manipulation is usually done. Okay, that is about transgenic animals. Please have a look at them. You can perform insertion, you can perform deletion. Those are, I hope you will get used to the terminology. This is a nice picture, this is a nice one also, the about generation of a chimera and then what you do. This is the recipient female and vasectomized male. When these two mate, we will get a pseudo pregnant recipient female. That means it is physiologically feeling that it has conceived, and it will hormonal hormones also will be produced so that it will hold on to the embryos. Otherwise, they might not be held, they might be rejected, or they might come out. I just want to show the terminology that you'll use. Gain of function, loss of function. Gain of function is transgenesis, or you can say it as knock-in. When you delete a gene or you insertionally inactivate it, you can call it as, usually it is a deletion, you call it as knockout. When you are not deleting the gene, but you are somehow suppressing the gene expression, then you call it as knockdown. RNA interference is one of the ways that you can do. Now we'll also have CRISPR based ones where you can suppress the expression of the of some of the genes. You also need to know what is meant by spatial and temporal. All these experiments will all, any experiment is usually whether you're going to follow the effect of that particular gene on a time scale over time or age, what is happening, then it is meant by temporal. If you're looking at the function of a particular gene, and if it is, what is it doing in the brain, and what is it doing in the a leg, then you're looking at it as spatial terms. Okay, if you have any questions, I would like to answer. I'm not including transgenic animals for tomorrow's exam. I thought I will, but I think it might be unfair. Any questions? Okay, prepare well for tomorrow's exam, and we'll see you at the time. Yes? Tomorrow we have 3:15. If you come at 3 o'clock, it will be better because I have a class right after that. Yes, it is at 3:15, right?
Yes, sir.
Okay, good. Try to come at 3 o'clock. We'll try to finish it little earlier so that I'll go for my class at 4:15. Yes?
Sure, sir. Also, could you
Oh,
please
okay.
confirm the subtitles that we'll have to look into, sir, for the test?